Hello my dear students, welcome to the MS Science Academy. Myself Minu Srivastav, and today we are going to discuss about the evolution of disinfective agents. Okay, so what is the evolution? Evolution of the disinfectant, it means it's for checking the ability or efficacy of any disinfectant, that how much this given disinfectant is effective, up to how much extent it will effective. Okay, there are some parameters or some methods to by which we will determine the effectiveness or efficacy of the disinfective agents. Here the couplet method, or we can say the tube dilution method. Second is the agar plate method, the tube dilution method also named as, and second is the couplet method. Third is the phenol coefficient, and in the phenol coefficient we will determine the phenol coefficient by two method, Riedel-Walker and Chick-Martin test, and fourth one is the Kells-Sykes test. First is the agar plate or tube dilution method. In this method, the liquid medium, this test is performed in the liquid medium like nutrient broth, or in the solid medium like the nutrient agar. In the both type you can do this. Okay, here first you will take the liquid water soluble substance disinfectant at the different concentration. Suppose that you will take the test tubes. Here the test tubes, 5 test tubes you have taken. Okay, and this test tubes, here you have taken the 5 test tubes. Okay, and the the disinfective agents you have added, that 6 ml, 5 ml, 4 ml, 3 ml, 2 ml. Okay, like this you have added different concentration. Okay, now the microorganisms are added at a particular amount. Okay, in all the microorganism concentration is same in all test tubes. Okay, and then give the specific time. After the specific time interval, these tubes are transferred into the nutrient broth, or if you are not prepared the nutrient broth as a liquid medium, but if you want to transfer, you will transfer into the solid medium on the agar plates. Okay, on the agar plates, or you can transfer the medium into the nutrient broth, okay, as in liquid form. Now the incubation takes place for the third. If the bacteria, if you have taken the microorganism as the bacteria, you will incubate 37 degrees Celsius, and if you will take the fungi, you will incubate 20 to 26 degrees Celsius. Now you will determine the minimum inhibitory condition. What is the minimum inhibitory condition? It means, suppose that in the 2 ml the turbidity is observed, okay, in so much amount, and in the 3 ml less turbidity, but in the 4 ml, 5 ml and 6 ml no turbidity or no growth is observed. That means the 4 ml is the minimum inhibitory condition by which the given disinfectant is inhibit the particular microorganism. Okay, so that is named as the minimum inhibitory condition, concentration, or MIC. Okay, like this we will determine. That's the second method, that is the couplet method. In the couplet method, nutrient agar plates are prepared. Okay, nutrient agar plates are prepared. Then the 0.2 ml of known concentration of inoculum is spread. Then in that after the spreading the inoculum, that cups are prepared. Cups are made. How the cups are made? We will scoop out the that agar. And just small cavities we will prepare by the help of sterile borer. Okay, and then 0.2 ml agent, the given test agent, is poured into all cups. Okay, now incubate for the 37 degrees Celsius for 24 hours. Now the zone of inhibition is observed. What is the zone of inhibition? It means the area by which the given disinfective agent inhibit the growth of microorganism. The zone of inhibition here 1, 2 and 3. Here the 1, 2 and 3. 1, 2 and 3. But the concerned zone of inhibition is in the 3 is more. Okay, so here the zone of inhibition is more, so that this in particular disinfective agent is more effective as compared to first and second that disinfective agent which are using. Okay, so here in the couplet method we will determine the we will observe at a time more than one disinfective agent. Now we will discuss about the phenol coefficient and the Kelsker test. Now we will discuss about the phenol coefficient test. In the phenol coefficient test, we will determine the disinfectant efficacy or the evolution of the disinfectant is comparable with the phenol. Okay, here the phenol is taken as the standard and our test disinfectant is compared with the phenol. Okay, that is why the phenol coefficient we will determine. But the method is the Riedel-Walker and the proper broth is the Riedel-Walker broth. So that term is the Riedel-Walker test or RW coefficient we will determine. Okay, but the same thing, the phenol is taken as a standard. The microorganism is used just like the Salmonella typhi, like Staphylococcus aureus, like the Pseudomonas aeruginosa, like type of the microorganism we can use. But the broth is the Riedel-Walker broth we will use. Okay. How is the procedure? The procedure, first the test disinfectant is taken and the phenol is taken. For the each at a fixed concentration the stock solution is prepared. Okay, for test disinfectant and for the phenol the stock solution is prepared with the fixed concentration. Suppose that the 10 ml of the disinfectant is dissolved in the 1 liter water. Okay, so that is the stock solution. We have the stock 1 liter and with the help of that 1 liter stock we will prepare the dilutions. Okay, so here the test disinfectant, after the stock solution prepared, we will have we will prepare the dilutions like 1:1000, 1:1200, 1:1400, 1:1600. Same thing we will do so with the phenol. Like after the preparation of the stock solution, we will do or we will just dilute in a different dilutions like 1:100, 1:120, 1:140 and 1:160. Here the main thing is that how the dilutions are prepared. So I will give one just a small idea here. If you want to prepare that 1:100 dilution, okay, for that you have to mix that 1 part of the sample is to 99 part of the that reagent or the solvent system. Okay, now the 1 part sample is dissolved in the 99 part of the solvent. So if you will take, it's a 10 microliter, so you will dissolve in the 990 microliter. So 10 microliter sample or test disinfectant or phenol dissolve in the 990 microliter of water. Okay, so here the dilution like this you will prepare of the dilutions or you can calculate the dilutions. Okay, now what we'll do, that Riedel-Walker broth tubes are prepared and that we will take the Riedel-Walker tubes are prepared, and in that Riedel-Walker Riedel-Walker broth, in that Riedel-Walker broth we will add that 0.2 ml 24 hours of the microorganism. Okay, now here different dilutions, in the different dilutions one point is remaining here, that in the different dilutions are added in Riedel-Walker, है ना, dilutions are added in Riedel-Walker broth tubes. Okay, dilutions are added in the Riedel-Walker broth tubes. Okay, now the 0.2 ml of 24 hours culture of microorganisms are added. Okay, 0.2 ml culture of 24 hours. Okay, 24-hour culture of any microorganism we will use. Then here just when interact with that when the dilutions is added in the Riedel-Walker tubes and that 0.2 ml 24 hours of microorganism are added, then the test tubes remain 2.5 minutes and go for the incubation. Again in second test tube go for the 5 minute, just keep stand with the interact with the 5 minute and go for the incubation, like this 7.5 minutes and 10 minutes. When we are adding, we just interacting with this test tubes, the temperature is maintained 7.5 to plus minus 0.5, means 17 and 18 degrees Celsius we will maintain the temperature, 17 and 18 degrees Celsius. Now like this chart we will get. Now after the incubation, examine the growth. Here the what kind of the growth or chart we have observed, like in the red no growth and the green which I have shown with the help of green, so growth is observed. Okay, so here how the Riedel-Walker coefficient is determined, that dilution of test disinfectant killing in 7.5 but not in 5 minutes. Okay, so in that solution, in that particular dilution, when the microorganism kill in the 7.5 minutes but not in 5 minutes. Okay, so here you will observe that 1:1200 dilution, that the microorganism kill in the 7.5 but not in the 5 minutes. Same as like in the phenol, the microorganism kill in the microorganism growth observed in the 5 minutes but not in the 7.5 minutes. So you will take the just divide like these dilutions and you will receive the 10. Okay, 1200 and 120. So you will receive the 10. The Riedel-Walker coefficient is 10. If the Riedel-Walker coefficient or the phenol coefficient is more than 1, that it means it is more and more effective than the phenol. Okay, it means it's a 10 times more effective than the phenol. But the Riedel-Walker coefficient has some disadvantages, or we can say some drawback of with this test. How is it so? That it is a just virtual coefficient, it's not realistic. Why I'm saying like this? Because in the hospitals there are just different kinds of the organic material, like fecal material, like organic material, urine. These organic materials are very much in a very high quantity in the hospital areas. Okay, so we have to observe our disinfectant or the given test disinfectant is effective on that particular organic material or not. Okay, so we can say it's a virtual. Okay, here, and the next thing is that it only one microorganism we are using, so we cannot determine that how it will affect on the other microbial flora. Okay, and the time duration is very slow, 2.5, 5, 7.5 and 10. So it's less effective for because the time duration is very short to interact with the microbial system and the disinfectant. So for the slow disinfectant, it's not that much realistic. Okay, so now we will discuss about the Chip-Martin test, because in the Chip-Martin test we will use the organic materials. With the help we can somewhat justify our procedure. Now we will discuss about the Chip-Martin test, that is more realistic as compared to Riedel-Walker test, because we will use in this the dried human feces or yeast we will use. Okay, here it is a modification of the Riedel-Walker test. The Charles Jam and the Harriet Chick is just developed this technique or developed this method in 1908. Okay, the microorganism is used is the Salmonella typhi, or we can use the Staphylococcus aureus. Okay, the procedure is that the 3 gram organic matter, either the dried feces or the yeast, is in 3 gram taken and mixed with the 10 ml water. Now it mixed with the 2 ml microorganism. The microorganism is previously prepared of the 24-hour culture we will use here. Now for different concentration we will prepare. Now in this chart I will explain the different concentration: 2%, 1.8, 1.6 to 1.45. like this concentration we will prepare, but for the each concentration we prepare 2 tubes each for each concentration. Okay, like phenol we will use and like the test disinfectant we will use. Okay, now for from this material or from this mixture we add the 2.5 ml of each tubes. Okay, which contain the different concentration or different dilution you can use. Okay, now allow to contact with the 30 minutes and duration, the temperature in this duration, the temperature around 20 degrees Celsius. Now in this mixture take 1 loopful of the reaction mixture and transfer the 10 ml transfer into the 10 ml recovery media. Okay, in this test tubes the 10 ml recovery media is previously prepared, means already prepared in such kind of the tubes, like in such kind of the tubes, and incubate 37 degrees Celsius for 48 hours. Now we will observe the growth, and after the growth observation we will determine the CM coefficient. CM coefficient is the mean of highest concentration showing growth in both tubes and lowest concentration preventing the growth in both tubes for same disinfectant and phenol also. Here Now I will explain that in the phenol, two concentration are highest, 2 and 1.8, but in the two highest concentrations, we will select the lowest concentration, which will not show growth, okay, or prevent the growth. So 1.8 is the lowest concentration which prevent the growth, so we will take here. Like this, 0.47 and 0.41, the 0.41 is just preventing the growth. Now the 1.62 and 1.45, the lowest concentration, but from the lowest concentration you will select the highest one, that is the 1.62. But here one is the loophole, that in the 1.62 showing the growth in tube 1 but not showing the growth in tube 2. So we have to take on the 1.41 here. But in the test disinfectant 0.37, 0.33, we will take the, in the lowest concentration we will select the highest concentration which are showing the growth. Okay, so here the 0.37 is taken. Okay, so we will take out the mean. Okay, here 1.8, 1.45, we will take the mean, and again we will take the mean and divide it, and the CM coefficient, that is the 4.15. It means that given test disinfectant is 4 times more effective than the phenol. Now we will discuss about the next method, that the Calske-Sykes method. Okay, now we will discuss about the last method, that is the Calske-Sykes test. In this method we will just interpret the result in the pass or fail term. Okay, that in this method we will not determine the inhibitory condition in zone of inhibition or minimal inhibitory concentration. We just determine that the sample is pass or fail. Okay, so what is the procedure? Now we will discuss, but before going through the procedure, we want to say that the synonym of this test, that is the triple challenge test. This test is also termed as the triple challenge. Why is it so? Because in here the 3 times of the microbial system, microorganism concentration are increased 3 times. Okay, here microorganism concentration is changed 3 times. The duration is generally 30 minutes, and the microorganisms are used as Pseudomonas aeruginosa, Pseudomonas vulgaricus, and that Staphylococcus aureus, E. coli. Like such type of the microorganisms are used in that Calske-Sykes test. Okay, the result is pass or fail term. The procedure is that the test disinfectant, just 3 ml we will take the test disinfectant, and in one test tube the microbial cell 1 ml is used, and we will interact with the interaction time, that is the 8 minutes, and after 8 minutes just we will take 1 loopful of that culture mixture or microorganism mixture with the test and disinfectant mixture, and spread on the 5 Petri plates. 5 Petri plates we will already prepare in the lab in the sterile conditions, and in the Petri plates we will keep the nutrient agar. Okay, so here the nutrient agar is used. Okay, and 1 loopful that microorganism and test disinfectant solution is spread, and after that incubation takes place for 24 to 48 hours and observe the growth. Okay, now in the second step, again 1 ml extra microbial cell is added, and now again 5 Petri plates are prepared with the nutrient agar, and that 1 ml microorganism when added, just keep it for 10 minutes. Then 1 loopful is added with the agar plate. Okay, and incubation. After incubation we will see the growth of microorganism. Now add again 1 ml more microorganism and keep 10 minutes. Okay, again in the third step we will add the 1 ml more microorganism and keep for 10 minutes. Now again 5 separate Petri plates are prepared with the nutrient agar, and transfer to the nutrient media, and check disinfectant kill the microorganism or not. Okay, so here the result is interpreted in pass or fail term, and why and how we will discuss and how we will interpret the sample is pass or fail. Now let us see here, if in the first and second step, if in the all three steps the all Petri plates have no growth, then it's a pass symbol. But for at least pass result, in the first and second step at least 3 Petri plates have no growth. At least 3 Petri plates have no growth. Okay, so like that we will pass the given test disinfectant, and if there is no such kind of the stage, like the in the first stage the microbial growth will observe, then that sample will fail. Okay, so I hope you have enjoyed the lecture. Any doubt, kindly ask me in the comment section. Otherwise, please like, share and subscribe. Thank you.